Hey guys, how are you? Welcome back to One Stop Biology. So today we are going to start off a very new chapter of class 12th NCERT Biology, chapter 2, which is nothing but sexual reproduction in flowering plants, right? So basically in the first chapter we covered in general reproduction in organisms wherein we saw asexual as well as sexual reproduction in various plants and animals, right? So now taking it further, what we are going to do is we will study in detail sexual reproduction in plants as well as animals. Okay, so we will start off with sexual reproductive reproduction in flowering plants specifically, which are basically angiosperms. Which are nothing but angiosperms. Okay, so basically we have seen a lot of flowers in our life. We have various flowers in our garden as well, like roses, or marigold, or a lot of flowers, right? And we use it very frequently in various phases of our life. We use it in conveying various kind of feelings, human feelings, like maybe grief, mourning, love, affection, right? So, what are these plants and what exactly do they perform? What's the function that they perform in their life cycle exactly? So, as you can see, this is a diagram of a flower, right? This is a section of a flower basically. And here, as you can see, that there are various parts. Right. So what exactly are these parts? So as you can see, this yellow part is nothing but the petal. So you can see that petals are basically the part which has a lot of colors in different plants, right? Now the other part is sepal. This is usually green. Okay. Now then after that, apart from this, you have the male reproductive part of a flower, which is nothing but stamen. Now that is divided into anther and filament. So if you see this structure, this is nothing but stamen, which consists of anther and filament. Okay, and the female reproductive part is this one. Okay, so this is the female reproductive part. Now that consists of stigma, where the pollen grain lands, and you have style, and then at the end you have nothing but ovary. Okay, so this is the basic section of a flower and various parts that they have. So what we will start after this is the pre-fertilization structures and events in case of flowers. Okay, so basically, yes. So basically, what happens is much before the actual flower is seen on a plant, the decision that the plant is going to flower has already taken place, right? So there are several hormonal and structural changes which are initiated to lead to that differentiation and further development of that flower. Okay, so inflorescences are formed which bear the floral buds, and then the flowers are formed. Now, what happens is in the flower, the female and male reproductive structures, the male structure being androecium and the female reproductive structure being gynoecium, that differentiates and develops as well, right? So, first of all, what we will see is first we will see the male reproductive system, the androecium part, which is stamen, microsporangium, and then the pollen grain as well. Okay, so basically you'll see that there are two different types. One is this, as you can see, both these structures. This is a stamen. Okay, so basically this is the male reproductive part of a flower, And then there is another structure which is a section of an anther. So because it is important because it has the pollen grain. So we'll see both these structures in detail. Okay. Now as you can see here that stamen has two parts, right? There is a long and slender stalk which is nothing but filament. So here in the structure as well, as you can see, this long slender part is nothing but the filament. And then there is a terminal bilobed structure which is the anther. Okay, so what you see in the whole structure is that the proximal end of the filament is attached to the thalamus of or the petal of the flower, right? And then the number and length of this stamen. So the number and length of this part basically is variable in different species of plants, right? And if you arrange it in different, say if you collect different stamen of different flowers, you'll see that they vary in everything. They vary in sizes, right? They vary in shape, they vary in numbers, right? So basically a typical angiosperm anther, if you talk about a typical angiosperm anther, then we see that it is bilobed. It is what? It is bilobed. What does this mean exactly? I'll show you that as well. Okay, so here if you see the anther structure, you'll see that it is bilobed. So here this is one lobe and this is the other lobe, right? And each lobe have two theca. So there are two rounded structures in each lobe, right? So basically each lobe has two theca and that is why it is known as dithecous. Okay, and then there is a longitudinal groove inside. There is a groove that runs lengthwise separating the theca. So there are these grooves which are longitudinal and it separates both the theca. Okay, now if you see that the anther is a four-sided tetragonal structure which has basically four microsporangia which are located at the corners, two each in each lobe. Now in each lobe basically there are two microsporangia. So in an anther there are in total four microsporangia, right? Now these microsporangia develop and they become the pollen sacs. So here if you see inside are the pollen grains, right? So let's label it once. So basically these are nothing but the pollen sacs. Okay, this is the filament. We also call it as stalk. Then these are the pollen grains and this is the line which separates basically, right? This is the line of dehiscence. Okay, so if we see that the anther is four-sided tetragonal structure, right? And it consists of four microsporangia, which are basically located at the corners, two in each lobe. Now, this microsporangia develops and becomes pollen sac, okay? And they extend longitudinally all through the length of an anther and they are completely packed with nothing but pollen grains, right? They are packed with nothing but pollen grains. Okay, these are nothing but pollen grains. Okay, now what exactly is the structure of the microsporangium? So let's see that as well, right? So basically, if we see a transverse section of a microsporangium, what you'll see that it is surrounded by first four walls. So what it is done is it is surrounded by four walls, okay, and these walls are nothing but epidermis, Then you have the middle layers. You have tapetum, and you have endothecium. Okay, so these are the four layers: epidermis, endothecium, middle layers, and tapetum. Okay, and inside you can see that there are microspore mother cells. Okay, so basically each microsporangium is surrounded by four wall layers. Okay, now the outer three wall layers perform the function of protection and help in dehiscence of anther so that it can release the pollen grain. But the innermost wall layer, which is nothing but tapetum, that nourishes the developing pollen grain. So remember that nourishes the developing for pollen grain. Okay. Now cells of tapetum basically possess very dense cytoplasm and they have more than one nucleus in general, right? So when the anther is young, a group of compactly arranged homogeneous cells, which are called the sporogenous tissue, occupies the center of each microsporangium, right? So if I just label this diagram, which is nothing but a young anther, what you will see is that this is nothing but connective tissue. This is epidermis, this is endothecium. Now these are the sporogenous tissue. Okay, and then you have the innermost layer, which is tapetum, and you have middle layers as well. Like, right? So basically there are sporogenous tissues present which occupies the center, as you can see, the center of each microsporangium, right? So basically this is the structure of a microsporangium, right? Okay, now what exactly is microsporogenesis, right? So every anther develops into sporogenous tissue, and the cells of those sporogenous tissues undergo meiotic division, undergo meiosis to form microspore tetrads. Now, what exactly these tetrads are, that we'll see in the next video. So just wait for that, and we'll cover the entire microsporogenesis and then the pollen grain, its structure, and what it has exactly, and which basically forms the half of the zygote when the fertilization occurs. Okay, so we'll cover all that in the very next video. I hope you understood the basic structure of stamen, what exactly microsporangium is, and then we'll move on to microsporogenesis. If you have any doubt, please let me know either in the comment section, or you can also message me on the WhatsApp number which is given in the description of the video. And if you do understand each and everything that I told you in the video, please like it and share it with your friends, right? And please do ask everyone to subscribe to the channel as well so that you guys can get all the updated videos notifications as soon as I upload it, right? Okay, guys, thank you. Bye bye.